A Punnett square is a diagram used to show simply the cross. Here a Punnett square is shown. At the top here we have the parental genotype, the first parental genotype, in this case homozygous dominant. And here you put the second parental genotype, in this case homozygous recessive. You then look down and across at each one. You combine them, so this would be a big A combining with a little a, gives us the heterozygous big A, little a. Similarly, down and across gives you big A, little a, and so on. These are then the offspring genotypes. In the example of a monohybrid cross, we're considering just one trait. So let big R stand for red flowers, so that's the dominant trait, and little r stand for white flowers, that's the recessive trait. This means that if it's homozygous dominant or heterozygous, the flower will appear red, but if it's homozygous recessive, the flower will appear white. So what's the outcome if we cross a red, pure breeding red with a pure breeding white? We can use a Punnett square. So we first of all draw our grid. We next fill in the gametes around the edges. Now we fill in the offspring. And that gives us our final cross. So pure breeding red, pure breeding white produce all heterozygous offspring which are red in colour. So what if we then take these heterozygous offspring and cross them with each other? Again, start with a grid, add in the gametes, in this case it's the heterozygous gametes. Now fill in the offspring, again looking down and across each time to fill in the offspring. Notice here how we always put the dominant allele first if they're heterozygous. So what we have now is 3 genotypes, homozygous dominant, 2 heterozygous, and 1 homozygous recessive. These 3 here will all be red, and this 1 will be white, giving us 75% red to 25% white.